potency of the antibiotic, it would die. But by letting one mutation be able to breach into the higher concentration of antibiotics, now that one can survive. So it's just like the lawn example. So now that bacteria, because it is resistant to that antibiotic, it can survive, and then another mutation happens to give it like a thicker exoskeleton to make it a little bit more apt to survive. So I'll talk to you about specifically, we're not talking about exoskeletons because we're not talking about bugs, but what are the genetic mutations that happen in bacteria to resist antibiotics? So let's give the beta-lactam example. So just make sure that you understand this beta-lactam structure because it's going to get more complicated. And as long as you understand like where it's coming from, so remember the beta-lactam ring comes from the penicillium mold and it blocks the active site of the enzyme within the bacterial structure so that that bacteria cannot lay down new peptidoglycan and it can't reproduce. Yeah, so it can't go on and perform binary fission. It can't lay down new peptidoglycan at all because the enzyme doesn't work, because it's blocked. Okay, so there's the bacteria. Usually the penicillium will kill the bacteria in the same way that our antibiotics will kill the bacteria in the same way. But now we have another way that, so now this bacterium is going to go through some sort of genetic mutation just like our bug did with the exoskeleton. So what happened was there was a genetic mutation somewhere along the line, and this bacteria, that one's already dead, so let's give you another example. So this bacteria had a genetic mutation that formed an enzyme that will cut, that will cut the beta-lactam ring. So this beta-lactam ring is now broken. So it can break that beta-lactam ring, and now that beta-lactam ring won't plug the active site of the enzyme anymore because of this genetic mutation that happened. Does that make sense? You guys still with me? Beta-lactam ring normally will kill the bacteria because it'll block the enzyme. But there was a genetic mutation. This genetic mutation was an enzyme that will go around cutting all these rings. Cut. Now the bacteria can survive. So now this bacteria has this enzyme that's called beta-lactamase. And it's an enzyme that breaks up the beta-lactam ring. So now it won't block this enzyme anymore because it got rid of the beta-lactam rings. It won't block this from laying down new peptidoglycan anymore. Now our enzyme can go lay down new peptidoglycan, and our bacteria is happy because it has this enzyme called beta-lactamase. With me? Yeah? Okay, because we're going to do this in lab, and there's one more step in the lab. So just make sure you understand this example, the example of how beta-lactams work to kill bacteria by destroying the cell wall. They destroy the cell wall because it blocks the enzyme called bacterial transpeptidase that lays down new peptidoglycan. But the bacteria that have this enzyme called beta-lactamase will break up all the beta-lactam rings. Now the beta-lactamase, the bacterial transpeptidase will lay down new peptidoglycan. Hopefully you're with me. Yeah?
How complicated is this mutation? Is it like in the production of the bacterial transpeptidase? Does it like during the production of that enzyme accidentally produce the aminase, or
so it's, mutations are weird that way because there was at one point one bacterium at one point had some sort of mutation, and I'm not 100% sure the origin of this original protein, but it was originally a protein. Usually how it happens is there's a protein that does something else, and there's a mutation, and that protein changes conformation, and now it can function as something else. So now it can function to break up these beta-lactam rings, and because it did that, then this bacterium by accident was able to survive and have millions of babies. Yeah? Okay, so it's just usually an accident that happens that makes a protein a little bit of a different shape, and now that protein actually can do something. Yeah, awesome. Yeah.
So does it it binds to the beta-lactam and it just cuts it, doesn't remove it?
Yeah, no, it just cleaves it. It cleaves the beta-lactam ring, so the ring isn't a ring anymore, and it won't fit into the active site of the enzyme anymore.
So then the transpeptidase just keeps going as normal.
Yep. Mm-hmm. Yeah. Perfect. Okay, so here, I am gonna have you guys do this on your own. So I'm gonna tell you a little bit about it, and then what I want you guys to do over the weekend is read this publication and just get an idea of what it's talking about. So I'm gonna try and start slowly introducing you guys to PubMed. Raise your hand if you've used PubMed. Yeah, probably a lot of you. Okay, so PubMed is like the Google for scientists. So what you're gonna do is you can click on this, or you can just search by that ID and it'll pull up the article. I think the article's free anyway. Let me just click on it and see if the article's free. Oh yeah, so it's a free full text. So, but I think you could probably get through just by reading the abstract. Oh, it's just a little Q&A paper. Okay, so this is originally the paper that it talks about antibiotic resistance and how it happens. So just read through that gently. Like, I'm just gonna read it just like I'm like lazy reading. Read through it and see what you think. And then I'll talk about it. I'll ask you something from the quiz, but from the quiz it's gonna be really straightforward. So it's not gonna be any of those tiny details, just straightforward kind of what these antibiotic resistance methods are. So one antibiotic resistance methods we've talked about is the formation of an enzyme that will break up the pharmaceutical. So remember, these come from the penicillium mold, but now we've taken these and put them into a pill, and in that pill, which is penicillin has all those beta-lactam rings in it. That's what penicillin is. So the person with the infection takes the pill. Now the bacteria will die unless it has this enzyme. That's its method of antibiotic resistance. Yeah, so here's our methods of antibiotic resistance. We have efflux. This is the coolest thing, I think. Efflux sounds way cool to me. I mean, it's cool coming up with your own enzyme that will kill something, right? That's really cool. But efflux trips me out because what they did is just create this like little vomit pump. So when the pharmaceutical gets in, when the drug gets in, they just spit it back out again. Like, how cool is that? There's a protein there was a mutation in a protein, and that mutation in a protein, here's I'll draw another bacteria for an example. And we give it some sort of drug, and it's just gonna spit the drug right back out again. So it made some sort of protein. There was a mutation in a gene somewhere that formed a protein, and that protein is an efflux pump, and it will just spit that antibiotic right back out again. Cool, right? So that's a method of antibiotic resistance. Immunity and bypass. So I'll have you guys read about what immunity and bypass means. So basically what they're doing is they, let me talk about the other ones first. So target modification is is target modification is when, let's draw another example of a bacterium. And here is a 70S ribosome. And that 70S ribosome, there's a 50 and a 30S subunit where if it just changes them a tiny bit, if there's a little bit of a chemical change in those subunits, now the pharmaceutical can't recognize it anymore. So it changed its target. It changed the ribosome or changed the topoisomerase in a way where the drug can't target it anymore. So that's target modification. Inactivating enzymes, so that's what this is. It's an enzyme that inactivates. So immunity and bypass, so what it's doing is basically creating like a, I'm trying to think of a good analogy. So for example, if it creates, if the target is the 70S ribosome, and there's an area of the 70S ribosome that the pharmaceutical will target, it's going to make a target somewhere else that looks like that 70S ribosome. So it's going to target something else instead. So it's going to like pull the pharmaceutical away from its main function.
So is it like a dummy?
Yeah, like a dummy. Yes, exactly. Like look over here, and then it attacks that, and then there's no pharmaceutical left to kill the ribosome itself. Yeah.
Sorry, and what's changing the ribosomes and the target modification?
Yeah, so in the target modification, so here in target modification, these are some examples of bacteria that have changed whatever these targets are. So if, I don't want to give you an exact example because that's just going to confuse you. So there's the one method is creating a pump. That pump spits out the drug. That's one method of antibiotic resistance. Another method of antibiotic resistance is creating a dummy. So like it's if the target for the antibiotic is this enzyme, creating an enzyme that looks kind of like it. That's very similar. So that's another option. Another option is target modification, so changing it. So if the active site is over here, maybe the method of antibiotic resistance will be changing to where now the beta-lactam ring comes over on this side. Now it's free to do its job. So it's changing the target.
Changing the shape of the target?
Yeah, it really is, because it's a chemical and it's changing the chemical structure. So it is changing the shape essentially, or the chemical itself. Yeah.
This is just theoretical, but if the bacteria can mutate so quickly, can the fungi not mutate quickly enough to keep up with it? Like, I mean, if you just put the fungi and the bacteria together, wouldn't the fungi be able to mutate enough to overcome these things?
Yeah, so there are natural methods of self-defense I'll talk a little bit more about that in lab, about the cat and mouse, where the penicillium is the cat and then the bacteria is the mouse, and then they keep changing. So they do, because they do mutate so rapidly like that. But the problem why we've put it on hyperdrive is just because of the amount. So the reason that mutations are way more rapid than they used to be is because of how much we're trying to kill bacteria all the time.
Well, the problem is we're not taking, I don't know if this is a problem, but it's not like we're taking the fungus. It's not like the fungus is actually in contact with the bacteria when the bacteria are changing, because we're just taking the chemical that the fungus made.
Yes. So they do have methods of defending themselves against the bacteria, but all of these methods have come like 50-fold just because the bacteria now, they've always mutated so rapidly, but when we target one specific thing, we're wiping out everything and giving that one with the mutation the opportunity to survive. So it's like it's like we're shoving everything into an Arizona Superdome where we're like, fight it out. You know, we're trying to like shove everything in there so that everything's trying to fight at a rapid rate. I don't know if I explained that well, but okay, so then the last one was inactivating enzymes. So it will inactivate enzymes and we can undergo antibiotic resistance in that way. So read through this paper just gently and see if you can understand it, but then also meeting with Dr. Welsh would be a really important one for this one because he can explain them one more time. I don't want you to memorize any of these antibiotics underneath the methods. I just want you to understand the foundation, what that fundamental method, like what each method means and how antibiotic resistance works in that way. Okay, so remember efflux and what that means. Remember immunity or bypass, what that means. Target modification and activating enzymes. So this article will really help you with that. Okay. So here is the antibiotic targets, how they resist, our cat and mouse game. So we're always trying to play cat and mouse. If the bacteria comes up with a way to resist the antibiotic, now all the drug companies are back in it, going, what do we do? How do we kill this next mutation that happened in the bacteria? What was the mutation? How can we add how can we make our pharmaceuticals more effectual based on what the mutation was in the bacteria? So it's just a constant, never-ending game of cat and mouse. So something that's important for us to think about is antibiotic stewardship. So you'll hear about this over and over in a clinical setting, but antibiotic stewardship is incredibly important. And so what it means is being responsible with the administration of antibiotics and with patients who take antibiotics. But we do have to have antibiotics, right? Like we don't want to die from a cut on the foot. We don't, people need antibiotics. My daughter had antibiotics when she was 3. She had 104 something fever and she had a spot on her lung, which we thought it was pneumonia. So, we were like, oh, let's give her antibiotics. So, her body probably could have fought it off, but, when you're getting up in those 104s with children, it gets really terrifying. So, even just the smallest things like a UTI, trying to control a UTI ourselves, but also, using your own self-judgment, but also definitely listening to your healthcare practitioner. And your healthcare practitioner, this is a whole thing as well. So my sister, she did a whole talk for us once on antibiotic stewardship. I can have her do it again if you guys want her to come and visit. But she's a physician, and it depends on the physician. Some physicians are like, it's lunchtime, here, take that, bye. And some physicians are like, I don't care if you are on death's door, I'm gonna think about it. You come back in 3 days. If you're still on death's door in 3 days, I'll give you some antibiotic. So it depends on the physician. But really, antibiotic stewardship is important, and it's really important to think about. Also, one thing I wanted to mention too is the environment. So why are there all kinds of antibiotics in the water off the coast of Florida? Somebody knows this. Why is it?
Is it urine?
Yes, urine. Why Florida specifically?
Old people.
Old people, yes, because there's a lot of old people in Florida, and they take a lot of pharmaceuticals. So, good stuff. Okay, so there's pharmaceuticals, especially antibiotics, everywhere. There's antibiotics in livestock. We use a ton of antibiotics in livestock. The majority of our antibiotics goes to livestock. So that's just something to think about. It's, it's not preventable unless we reduce our consumption of livestock, but because they get very, very ill, and they can get very ill fast, and then people can get ill from their illness, so it is important unless we reduce our consumption. This is a new slide I put on here because I thought it was interesting. Was out of 100 patients that were discharged from a short medical stay, 78% had antibiotic overuse. It's almost 80% are misusing antibiotics out of patients that are in the hospital. So, you know, we're caring more and more, just like climate change. You know, it's like, are we gonna care enough to do something about it before it's too late? And it's gonna be the exact same situation with antibiotics. Are we gonna care enough about it before it's too late and our antibiotics are completely ineffectual and we go back to the 1800s when we end up dying from getting a paper cut? Yeah.
That's not really the patient's fault. They
Yeah,
just
no,
do
it's
what they're
not the patient's
told.
fault. No, it's definitely not the patient's fault. Yeah, it's not. Well, you know, I would say it's not the patient's fault. Most of the time it's not. But also a lot of patients, and I have from my sisters that are all in the medical field, one is a doctor and 3 are nurses, that's what patients want. You know, they're coming in and they want antibiotics. And you can fight them, like that's what you have to do. That's what my sister does for the majority of her job, is say, no, you don't need antibiotics. Next patient, no, you don't need antibiotics. Next patient, no, you don't need antibiotics. So people come in because they want treatment, but a lot of times that's the part of what is important to me for you to get across in this class of why you would need antibiotics if it's bacterial, how to recognize if it's bacterial or if it's viral. And you know, really, a lot of people, 3 out of the 5 girlfriends that I have that are mothers, every single time their kid has a fever, they take them and get them antibiotics. Just all my friends do. You know, even after I tell them, like, maybe think about, you know, and also because for females, it wipes out your beneficial population. So even in the vagina, it's not healthy to wipe out your beneficial population, but also gastrointestinally for men and women, and even on your skin and your eyes. And you know, people get those super infections just because they've wiped out all their beneficial bacteria. So I'm not saying don't take antibiotics if you need it, but you know, take them responsibly. Be careful of like, okay, do I really need antibiotics? Do I not? Do I believe in my practitioner? Is my practitioner, you know, do I think my practitioner is making good choices or not? So it is definitely a huge healthcare problem, but also the patient can take some control of that too. Yeah, did you have a question? Okay. Yeah.
So going back to the slide where it has how antibiotic resistance happens. The first like little circle.
This one?
Is it? Yeah. Is it saying? Oh no, sorry. It's just like the little cartoon of bacteria. But is it saying like there was already a bacteria that had the mutation, or is it like? Because from what I've like kind of understood about antibiotic resistance is that like maybe just didn't kill all of the same bacteria and one was left over, but this makes it look like one was already mutated.
Yeah, that's a fantastic question. So I'm gonna restate your question so everybody can hear that because it was important, but let me know if I'm not saying it correctly either. So there are a couple options. So one option is, this is your patient's intestines and they have an infection, they have an E. coli infection, it's all over in their intestines. So they take antibiotics and it kills off this many because they didn't finish their prescription. So then there's this many left. They're starting to feel stronger for a couple days, they feel a little bit better. Now these bacteria have the whole meal again, and then they get it again. So that is definitely how antibiotic resistance can occur. Let me just go along with that. So that's what happens with antibiotics. That's a big problem. Another problem with antibiotics is you kill, so your patient has a viral infection and your patient has a viral infection, they want antibiotics, they take antibiotics, it kills all of their beneficial bacteria. Now they have an infection because it was viral. That's another option. Another option is your patient has an infection. Now they've taken antibiotics. There's one that had a mutation at some point. They've taken the antibiotics, but the antibiotics don't work against this bacteria. So you've killed off all the ones it does work. Now you have a population that antibiotic is ineffectual. So now you have to change antibiotics or stronger antibiotics or a different you try different things to try and this is generally what happens. So this is the reason why generally this is what happens is I wouldn't say generally this is what happens. Generally this is the problem. Because now your patient has an infection that can't be treated or is very difficult to treat. Yeah, so I just got an email actually from one of my students last semester who had a staph infection, and she said, it was really nice, she just sent me like a super nice, like, I was so grateful that I was able to take your class because I knew the antibiotics they were giving me, I understood what my infection was about, I could correspond back and forth with the physicians, I knew what they were saying. So she felt more empowered by, educating herself, which was so great. But at the same time, what happened was she had some sort of resistant bacteria that had an efflux pump, that had a different enzyme, that had something going on where it had some sort of mutation, and now it took over. Then she's hospitalized because they're like, okay, let's try these super hardcore IV antibiotics. Okay, well, then she leaves herself exposed to C. diff and leaves herself exposed to some sort of MRSA, some sort of something else. So generally that is a huge problem, is what is happening is the mutation in the bacteria will take over and now they've got something else going on.
Is that mutation, is there some sort of cause from like a previous path that might have taken from some time ago, or is that it just like happened that there was one that was different? Like just
Yeah.
bad timing?
Yeah, so it can be within the patient. It's like the mutation can happen within that patient, but also the mutation can happen from a patient in a different room and it's spread to that patient, or that mutation happened and it's in the air or on a fomite and then that it was given to that patient. Does that make sense? Okay, everybody with me? Yeah.
Kind of off what she just said a little bit earlier on the slide that you just added, the one right above it. So when you say it's like not the patient's fault or it's the doctor's fault or who's really faulted in
Yeah.
this,
Mm-hmm.
are the doctors or whoever is prescribing the antibiotics, are they ever worried like in their mind like, oh, if I don't prescribe them what they're asking for and then something happens, are they ever,
Yes.
so they are, okay.
That's a
So they're
fantastic
rather
question.
over-prescribe it than.
Yes, rather over than under. That is a fantastic point. That yes, my daughter, she probably would have been fine without antibiotics. Her body probably would have fought it off. But does the doctor want to risk it? No, the doctor does not want that lawsuit, or even never mind the lawsuit, but the burden of somebody else's life in your hands is terrifying, I'm sure. So they are playing better safe than sorry, and some physicians, it's worth it for them to just blanket with antibiotics versus some physicians who worry about the long-term health of the population. So yeah, it's half a dozen of the other, you don't really know. Yeah,
So it's gonna be
great point.
pretty difficult for because you're talking about with like climate change and how this is kind of similar where eventually it's gonna catch up with the population,
Mhm.
but like everyone's kind of afraid because it's like there's obviously a balance you have to play.
Yes.
And it's kind of like a tolerance on one side where
Yeah,
if someone dies you don't prescribe them, then that's like a catastrophe for you.
absolutely, absolutely. And I think I from the very little clinical environments that I've been around, it's way more of the doctor says, I want to go to lunch. It's mostly that. it's mostly from my experience from working in a hospital for 7 years that they're like, sweet, I've got 20 more patients today, have fun, yeah. So a lot of times it is that, but you know, it is, yeah, they do wanna go to lunch, but also they do have 20 more patients, you know, and it's not an easy job, everybody knows that, it's a hard job, so, yeah, all the problems. Same as climate change, all the problems, we can't fix them all, but we'll try. Okay, so 100% of patients with COVID received antibiotics, it's just something I thought was crazy to look at. Of outpatient antibiotic prescriptions unnecessary, 30% are unnecessary that are prescribed. That's a huge percentage. Okay, so that's it for antibiotics and antibiotic control. How are you guys feeling about that? Yeah, and bacterial control, feeling pretty good about it, ish? Okay, so, this is our bacterial wrap-up. We're done with bacteria. Today's our last day. We'll be done. Not ever in the lab. We're gonna do that till the end. But in lecture, now we're moving on to viruses and protozoa and helminths, and you're gonna learn about some other organisms that make us sick. So this is, you know, we started with a huge concentration of bacteria, and the first part of the semester in my mind is the toughest part because you are learning some of those biology fundamentals like replication, transcription, translation, metabolism, like all of those big heavy hitters that we've had to learn. So hopefully from here on out is a little bit more, I don't know, maybe Dr. Rosebeer thinks it's, I don't know, did you, from here on out, did you think it was like a little bit easier, or did you?
No, it just got harder.
It just got harder? Oh, sorry, then never mind. Okay, well, hopefully you made it through that first part and you're feeling okay and we're moving forward. Okay, so this was just some quote that I found. 7.7 million people in 2019 died from bacterial infections that year. So about 7.8 million people die a year from bacterial infections. That is a lot of people. 8 million people a year die from bacterial infections around the world. That is a lot of people. It says that second leading cause of death globally after heart disease, ischemic heart disease, which is crazy because that's preventable, but heart disease, and then Staphylococcus aureus was the leading cause of death in 135 countries. Crazy. Okay, so let's wrap up. Bacteria are ubiquitous. They're everywhere. They live in all kinds of different environments. They can be aerobic, anaerobic, facultative. We've learned a lot, right? We've learned a ton about bacteria. We've learned their shapes. We've learned their colony sizes. We've learned a little bit about if they like to live in different temperatures and different salty environments. We've learned that they're absolutely everywhere, that most are beneficial. Most actually are, they go about living their life and they don't have nothing to do with us. Most bacteria on the planet. But according to the human population of bacteria, most, it's like 85% are beneficial. But there's that small percent that are pathogenic that we worry about and talk about and study because we're trying to prevent illness. And many people died until we figured out how to prevent, which still, I'm going to read you a book. It's on my to-do list to read you guys a book about kind of how that science came to be, that how we can treat open wounds and things like that. But still, it's a problem. Still, we have a hard time with hand washing and equipment washing and maintaining sterile environments. So how we treat them is with we prevent infection, we identify the organisms that we still are going to do a ton in the lab, and then we can treat them with antibiotics. Most people, or people that are still dying in large numbers due to exposure rates, lack of access to healthcare, comorbidities. So we're going to talk a little bit more about comorbidities when we get into the immune system. But a comorbidity is, like morbidity is disease, mortality is death. So sometimes you'll hear them together, morbidity, mortality. But morbidity is illness, So a comorbidity means somebody who already has like diabetes, for example, already has heart disease, already has congestive heart failure, already has COPD. Something is going on and then they get an infection because their immune system is down. So that happens. Misidentification is a problem. Antibiotic resistance is a huge problem. They come in many shapes and arrangements. We have gram-positive and gram-negative. Now we know that that's the peptidoglycan cell wall. We know peptidoglycan is what makes bacteria unique. They're prokaryotes, so they don't have a nucleus, they don't have membrane-bound organelles. The cytopathic effects, they have endotoxins and or exotoxins. So remember, endotoxins are the lipopolysaccharides, exotoxins are spit out, and those are type 1, type 2, and type 3. We treat with antibiotics, so we're learning about all of the antibiotics, but we treat bacterial infections with antibiotics, not viral infections, which we'll learn about all the different ways to treat all the other organisms that we're going to talk about. Antibiotic resistance occurs when there's a mutation that allows for the advantageous phenotype of that bacteria. So here I just put some in a nutshell. Here I put some information about gram-positives. Here I put some information about gram-negatives. So here you are going to have to know this for the, I haven't decided on the measure. The reason I'm trying to decide what's best for you, even though you might tell me what's best for you, but it's like my daughter with the broccoli. She will tell me what's best for her, do I believe her or not? So if we leave it all the way till the final, will you study it? If we make it where you study it in the midterm, then you have to study it again for the final. Let me think about it. Okay, but this is the mnemonic that I came up with. It's a silly one, but the reason I put them in this order is because, well, I didn't put them in any order for any reason, except these were the most common bacterial infections, and they're the most common bacterial infections that fit under the words, which was actually trickier than it sounds. So bacteria look like plums. So it doesn't all fit perfectly. And the reason I said bacteria look like plums, it's because all I can think about, because they look like little plums. Those are our cocci, gram-positive cocci. So the reason why I want you to know the diseases that are associated with bacteria is, again, because that's important for you to know as a layperson, as a human being, and as a clinician, to know what is bacterial versus what is viral versus what is fungal, all that kind of stuff. Yeah.
So if we have this in the picture behind, it would look like that, we would just basically fill it out.
No, generally what I've done in the past is like salmonella, and you have to put, is it bacterial, is it viral, or is it fungal? That's what I've done at the end, usually. So what I would do in this case is something along those lines. You'd just be able to have to like name 3 bacterial diseases, something like that. Yeah.
So what would you title each of the columns?
I wouldn't title, well, don't think of them this way, think of them this way. So think of them as rows. So I just put there are no columns. The reason that they're rows is just because those are the ones that start with a B. Bronchitis is bacterial, but it can be viral. I'll tell you the difference between viral and bacterial bronchitis. Botulism, bacterial vaginosis. So remember, botulism is Clostridium botulinum, has that exotoxin. You didn't have to remember that one, but just if it helps you kind of come up with, oh yeah, that was right, that was an exotoxin. And that was for Botox. Bacterial vaginosis, that was BV. That was on our note cards, was from Gardnerella vaginalis.
So I'm kind of confused about what memorizing this will do for us.
So what memorizing will do for you is when you get to the end and I say, what are you going to treat with the antiviral? Is it bacterial vaginosis? No, you have to treat that with an antibiotic.
Okay, so you would give us one of these and we would know they're one of the bacteria
So
symptoms or illnesses.
yeah, you would, so what you would do is when you say, is it tuberculosis? You have to recognize that tuberculosis is a bacterial infection.
Okay, so these are all bacterial infections.
These are all bacterial infections.
Okay.
Oh yes, sorry. Okay, make sure that that's clear. Thank you. Yeah.
How do you usually set up your midterm? Like, is it like multiple choice? Is it from like
Same as a quiz.
consistent,
Same as
just
a quiz.
longer?
Yeah, just longer. Same as quiz, just longer. Yeah. Okay. Okay, so these are bacterial infections. Otitis media is an ear infection. The reason I put it under A because there are no O's. So acute otitis media, that is actually bacterial, generally is acute otitis media versus chronic otitis media is generally viral. Don't care about that, but just that fits under A. Otitis media is an ear infection. Cholera, remember we talked a little bit about cholera. We talked about C. diff, we talked about chlamydia. Conjunctivitis is an infection of the conjunctiva of the eye. Tetanus, tuberculosis, tularemia. So we can go through these one more time before the midterm, but go through those on your own and just see if they make sense and see if you can kind of recognize where they're coming from or pull together. You know, if you do most of your studying from everything else, this will just be a couple where you're like, oh, I don't really recognize that. Where does that come from? Then you can pull that back in. And then when we get to the end, we have like 4 or 5 lectures that are diagnostic lectures that are only diagnosis. So then we're gonna look at a staph infection. What does a staph infection look like? Why does somebody get a staph infection? And then we're gonna look at pneumonia. Okay, pneumonia, is it bacterial, is it viral, is it fungal? What does pneumonia look like in your patient? So we're gonna go through the diagnostic section at the end of the semester. Okay, so here's a little blank chart if it helps you. Here's the organisms that cause it. You don't have to memorize any of these unless they're on your note cards. But the organisms, this just helps me sometimes. I don't know if it helps you, but you can look at the organisms that can cause enteric infections or intestinal infections, Mycobacterium tuberculosis, that helped us. So go through those. Just read through them. I think always reading through different genus and species of bacteria. Because as a clinician, if that's where you're going, then you'll see them come up again and again. Okay, so gram positives, gram negatives, those were our bacterial domains. And we are done with bacteria. Okay, so we're gonna do archaea and in about 10 minutes, and we're gonna be done with archaea. Okay, so we're done with bacteria. We can close the book except for studying, and we're moving on. So what are archaea? So do you guys remember, I don't know, this is gonna be like throwback to the first day of the semester, but remember why we don't talk a lot about archaea? Anybody remember? It's a tough one, because I said it like 100 years ago. Do you remember why we don't talk about them very much? Anybody throw a wild guess?
They don't make us sick?
Because they don't make us sick. Yeah, because we don't know of any archaea that make us sick. So why are we going to talk about them? Well, we're going to talk about them because as microbiologists we should, because they're part of our microbiology umbrella. But I'm just going to mention a couple things about them. One, that they're a prokaryote. So they are in the prokaryotic family. So what does a prokaryote mean? What does that mean?
Before nucleus.
Yep. No membrane-bound organelles, no nucleus. Awesome. So they're very simple, just like bacteria. We don't have to memorize any new structures or any new functions because they're the same. Same as bacteria. What is one thing that they don't have? Peptidoglycan. They don't have peptidoglycan. So they are the same, but they don't have peptidoglycan because that's what tells us it's a bacteria, right? So even though they are similar, they have similar structure, they don't have peptidoglycan. I will 100% ask you that. So everybody make sure you know that. So here's the prokaryotes. It's in this prokaryotic realm, not in the eukaryotes. They're very simple, they're very similar. They have prokaryotic ribosomes, so they do have 50S, 30S subunits. And I'm just gonna throw this out there, even though I'm never gonna ask you this question. that part of a ribosome, so ribosomes are enzymes. What are enzymes composed of? Proteins. And what are proteins composed of? Amino acids. Awesome. So protein, so it's just a big globular protein. And proteins are made up of a bunch of amino acids. So if you say there's a 50S subunit and a 30S subunit, it's actually a chain of amino acids that all bind together to form this big giant glob. Well, what's kind of funky about ribosomes is they have RNA as part of their structure. There's RNA in there, part that makes their big giant globule. So you're never going to be asked this. I won't ever ask you that. I just want to throw it out there. The reason that archaea are a little bit funky is because their RNA that's inside of their ribosomes are a little bit more like ours. So that's why they're in this weird part of the branch. This is why they come over here, is because their ribosomes, even though they're 50S and 30S, their RNA is a little bit more like ours. So they're off on their own separate branch. So I won't ever ask you that. You don't ever need to know that. Just know that they're a prokaryote. That they have prokaryotic ribosomes. They belong in the prokaryotic domain. This is where they are. So they're non-pathogenic, but that doesn't mean they don't live on and around us. So we have a lot of archaea that live on and around us all the time. They just don't make us sick, fortunately. So we have archaea that live on our skin, in our mouth, in our oropharynx, in our lungs, in our gastrointestinal system, in our urogenital system. We have archaea that live everywhere, just like bacteria. Yeah.
Do they compete with the bacteria?
I don't know that.
For resources?
I would guess. My best guess is yes, but I don't know very much about archaea. My archaea knowledge is extremely limited because most classes are geared toward human health, and so you kind of leave those out. You learn about them a little bit in bio class, and that's it. Yeah.
So, off just exactly what you just said, what kind of student or what kind of program would someone go into that studies archaea?
Yeah, you would be a microbiologist, and then that would be you'd have to go to a school that specializes in
Like a
that.
graduate program. That's
Yeah, in a grad program that specializes in.
How common? Like, do you know anyone that does that?
There are a lot of people that are working on archaea, yeah, but I don't know anybody personally.
like
There's
any-
probably, I would guess there's like maybe a team or two in like UCSF maybe, or a team or two.
What kind of
in-
like goals? Like, do they have? Like, what kind of what do they tell the people that fund them? Like, why do
Well,
people fund them?
So the reason people would fund them is because it's like exploring Mars. Like, why do we care about Mars? We can't live on it. People think we might be able to, but the potential, and archaea, maybe that there's some sort of pharmaceutical, and you'll be a multi-billionaire because you found something that archaea does that bacteria doesn't do. So there's potential in it. So that's, yeah. Most of science is just exploratory anyway. So they fund things that you can, that maybe will make somebody all kinds of money, and that's why they fund it. But also, universities like exploring. So yeah, it's a cool area of research for sure. Especially, I'm going to tell you why it's a huge area of research for environmentalists. Okay, so bacteria, archaea, eukaryotes, here's some similarities and differences. You can go through those. What is the problem? Why did I even bring it up at all? Well, one, we're microbiologists. It's good to know what archaea are in some sort of just 10-minute overview. But the biggest problem is cattle. This is why we have a huge problem with archaea. So archaea live in our intestines, and archaea live in the intestines of livestock. And cattle is our biggest problem with archaea. So you might not know it, but the reason there is like 80%, 70%, I would say probably 70%. I always throw out random percentages just that come off the top of my brain from something I read somewhere, so don't quote me, but it's a huge percentage, 70 to 80% of climate change is from archaea. Why? Because they live in the intestines of cows. And when they live in the intestines of cows, they eat the food that the cows eat, and they produce methane. as their byproduct. So some archaea, they're called methanogens, and they produce methane. So they produce methane as their byproduct. So, the bacteria that we talked about produce CO2. Well, there's some archaea that produce CH4, and that's a methane byproduct. And because methane is way more powerful that traps heat in our environment, then it's a huge reason why we have rising global temperatures, is because of the methane emissions produced by livestock. So cows, they have what's called a rumen. They don't have a stomach, they have what's called a rumen. I know way more about the cow's rumen than I would ever wish to know, because I went to Cornell and it's an ag school. But have you guys driven through like I-5, like driving down I-5? And when you go through the like factory farm area of the livestock, it's like, where is that? Like, I forget what that area that it is. It's like central California, before Bakersfield.
Fresno?
Yeah, it's Fresno-ish area. Yeah, and it smells terrible, but also the temperature is hotter because of the methane. So it's methane is a greenhouse gas. It is extremely effective at trapping heat and trapping the sun's rays. So what happens is, because of the archaea that live in the rumen, then they're just digesting and living the dream, and they produce methane, and because they produce methane, they breathe out CH4, which seems crazy that a huge problem with greenhouse gas emissions are literally burps and farts of cows, is why our planet is going down. So just, it's hilarious, but also extremely sad. Yeah.
So we haven't figured out a way to kill archaea?
No, well, they can't. They have to. They're a commensal that live in the rumen, and they help them digest the cellulose that they eat. So yeah.
Do they help us do that, or just?
No, they do. Also, we produce methane too, and we produce methane because of the archaea in our intestines as well. Yeah. Awesome.
Is there
Not
like
to the amount.
difference? Okay, yeah, not
Yes,
the amount.
yes, in access, it's like, I forget what the percentage is, there is a percentage compared, human methane production versus animal
Like,
methane
at the
production is like,
individual level, like
on the
versus
individual
like
level,
how-
way more, yeah, yes.
Does that do to diet, like something in their diet or something?
No, it's because they eat cellulose, so when we eat cellulose, some of the bacteria and archaea in our intestines can eat that cellulose and produce byproducts, but that is their main source of nutrition, is from eating grass and then, or hay or whatever it is, like those indigestible cellulose molecules that we can't eat. And then that's what the archaea eat, and that's what they break up those molecules and produce CH4. Awesome. Okay, so climate change, you may not have known it, but a huge problem is enteric fermentation. I've seen different, so when it's 70% caused by livestock, part of the problem is the area that livestock have to live on. So livestock have to live on a larger land mass of area than humans do. So it's also clear-cutted land mass and transportation to and from generally Brazil or Mexico, things like that. So there's a lot that goes into it, but I've seen different amounts. Yeah.
What is enteric fermentation?
Oh, here, I did have a slide on that. Enteric fermentation
Mhm.
is like, enteric is intestinal fermentation.
Okay, so that's specifically the cows, or cows and people and everybody.
Yeah, so enteric fermentation, so we have enteric fermentation also. So we have intestinal fermentation from bacteria also, or archaea. So remember, fermentation is just anaerobic metabolism or facultative anaerobic metabolism. Yeah, so we're like a very small percent compared to cattle. Okay, did you see that one? That one, I didn't realize that I had put that one up there. It was humans versus cattle, yeah. Okay, Archaea wrap up, that was it. They're ubiquitous, they're everywhere. They're probably the most abundant on the planet, which a lot of people say this, but because there are so many archaea that live in the oceans, difficult to cultivate in the lab. They're actually extremely difficult to cultivate in the lab, but there are labs that do have archaea labs. We usually isolate and study them by using their ribosomal RNA. They are none that we know are pathogenic to humans. We just figured out H. pylori in our stomachs. That's what it's a bacterial infection that caused stomach ulcers. We just figured that out maybe 10 years ago. So we don't know yet. Maybe it is, maybe there is something going on that we don't know of yet. They're harmful to the planet because of CH4, and they're prokaryotes. Okay, so we're done with archaea. Yay, we're moving on. And we're catching up, which I'm so excited about. This has been stressing me out. Okay, but we're moving on to viruses, and which is a perfect day because I woke up this morning with a giant cold sore. So have a cold sore because I'm talking about viruses, and never mind, it's like always when I'm the most stressed and it shows up on my face, and when I talk about something, I always end up getting it. So hopefully I won't end up getting any staph infection or anything like that. But we're talking about viruses today. I've been thinking so much about viruses. Now I have a cold sore. Well, I'll tell you what that is and how those happen. But now we're going to start talking about viruses. So bacteria, we've done a really good job kind of covering all the bases for bacteria. What I would do at this point is maybe, depending on what your weekend looks like, or reorganize your bacterial notes, kind of go back through those and like, okay, what are bacteria? What are all the pieces and all the morphology and the characteristics and everything that has to do with bacteria? Organize your notes, make them look really nice, then shut the book after you're done studying, put that away. Okay, we're moving on, so that your brain is not going in two separate places. So viruses are very, very, very different than bacteria. Very different. They're very different than anything. They're super weird and very different, and you'll kind of get a sense why. So what this is a bacteria that has a viral infection. So this is a bacterial cell full of viruses. Super weird, right? So this one bacterial cell has lysed or broken open because of all of these, the virus replicated over and over and over inside of the bacteria and spilled out. And that's what is going on when you have a viral infection or your patient has a viral infection. They replicate inside of cells so much so that they spill out, and then they can all go and infect new cells. Yeah?
Like some sort of viral infection, does it usually start as a bacterial infection that leads to a viral infection?
Great question, not usually. You only usually have some sort of co-infection if you're really sick or you have a low immune system or something like that. Generally, if you have COVID, you don't generally also have a staph infection unless you're in the hospital and you got a staph infection because you had COVID and your immune system was low. Yeah, great. Yeah.
Except for COVID and strep
COVID and strep. And what I'm guessing the reason is because of that's what's happening to the cells in your respiratory tract and leaving them open to bacterial infection.
Well, and
if
also
you're
because
around
the
bacterial
symptoms are
infection.
similar enough that you don't realize you need to go to the doctor.
That's a good point. That's a really good point too. Yeah. Did we get that right? Yeah, man, that is a fantastic question because a lot of people don't know, and a lot of even physicians don't know. So I'm going to give you a couple little tips and tricks towards the end, but always it's the best thing to ask your physician. But there are a couple little signs that it's viral and a couple little signs that it's bacterial. If there is a patient had an open wound and the patient had surgery and there's an infection lining the surgical site, it's bacterial. Viral, they have specific, do you remember that term tissue tropism? They have specific tissue types that they infect. So we'll be able to semi-recognize those, and we'll talk about some specific viruses. So, you know, if your patient has, let me think about it, like a UTI. Your patient has a UTI, it's E. coli, most likely. So then you'll know, okay, I can recognize if it's a urinary tract infection, it's probably bacterial. If it's respiratory or gastrointestinal, it could be either. So just a couple little tips and tricks about how to recognize some of those. But they're hard to recognize as well, and that's why we have tests. So that's why they do all the laboratory work to be able to see what is going on. Okay, so Pasteur is the one who made up the word virus. Well, he didn't make up the word, it's in Latin, but he's the one who coined the term virus to name this thing that he had no idea what it was. We didn't know what it was till a lot later than Pasteur, but he had a suspicion that there was something else going on other than the ones that they could figure out in the lab. Okay, because we can't culture viruses in the lab. They're too small, and we don't know how to yet. So I'll tell you kind of what viral virology laboratories do and how they culture viruses in a way, but we'll talk about that. So what is a virus? Super weird, right? This is my favorite. I love this virus so much because it's super alien. You know, if you were to say, oh, there's life on Mars, and they came out with this picture, everybody would be like, yeah, that's legit. That sounds about right. It's so alien and weird, right?
If they're so small, like, do you know what the magnification of that is?
Yeah, this is a computer model of what it would look like, but there are transmission electron micrographs, and it's TEM and SEM. What are SEMs? They're scanning electron micrographs now, and I'll show you some pictures of those, but this is like a computer model, yeah. But we know what all these structures are. We know 100% this is what it looks like, and this is what they do. But this is a specific type of virus, so we're gonna learn about the different types of viruses. This, fortunately, is only a virus that attacks bacteria. So this virus their tissue tropism, their tissue type is not human. They cannot attack human cells, they can only attack bacteria. So think about that. Yeah, go ahead, Dr. Duffy.
So do they like walk around?
Yeah, they do. They have like little, just like you would imagine. Yeah. I'm sorry, go ahead.
Well, I mean, obviously they don't like make decisions and anything like
Yes,
that, but like,
fantastic.
what kind of, how do they decide what to do?
Yes, very great. They're chemically attracted to things. So if they have chemical attraction to things, then the chemicals will come together, and that's it. It's just chemical.
Okay, so there's no level of higher thinking involved in
Yep.
that. Okay.
Yeah.
just
Such a good
making sure.
question. Yeah, no, you kind of have to like let your brain morph around it a little bit, because your brain, I'm gonna totally blow your brain away with this next few things, because you kind of have to go through that process, because viruses are so weird. So yeah, as we go through this, keep asking the questions, because they're all great questions. Okay, so this is a virus. This is a virus infecting us. The yellow is a human cell, and these blue things are all viruses. That's Ebola. That's what the Ebola virus looks like. This is COVID, or the coronavirus. This is a SEM or a TEM of the actual virus itself. So they can use electrons, they bounce electrons off of the virus so that they can tell what the three-dimensional structure of the virus is. So they can't actually physically look at it. It forms a picture by bouncing the electrons off the image. Yeah?
What magnification is it?
That is a really good question. I don't know what SEM and TEM they have up until this point. That's a really good question. I'm not sure.
Is that singular yellow thing
Yes, yeah, so this is the virus, this, and all of these little fuzzy things, that's the virus too. Yeah, so
Is it
I'll
all
show you
one
what,
organism?
what the shape is. Yep, this all right here, yeah,
Okay.
yeah.
Oh, that's not a human, that's not a cell.
No, that's the virus. Yeah, so I'll show you kind of what they look like. These are all viruses as well, so these are all viruses on the outside of these cells. Okay, so are they alive? So we know bacteria are alive. Ish. Like, do we think they're alive? I mean, they have DNA, they replicate, they eat. Why aren't they, right? They just don't have a nervous system. They don't have a digestive system. They don't have organelles, but, you know, they replicate. Yeah, maybe? We think so. Okay, so what viruses have are proteins surrounded by DNA. So if there are only proteins and only DNA, is that a living thing? Yeah, I mean, people say no, but after we go through the whole viral section, I have a hard time being convinced that they're not living, but you just have to form your own opinion. Yeah.
Don't biologists define life as being able to replicate without the aid of another organism?
Yeah, so I'll show you what the definition, like the official definition is. Yes,
So by
yes.
the official definition, no.
But that's true, but a lot of organisms as well, like there's certain types of bacteria that can only replicate inside of other things, or there's only certain types of fish that can replicate without the aid of something else, or like, you know, there's all of these blurred lines. But so, I mean, if you're talking about replication and you're looking at this, then why not? You know, like, so we'll go through it and just, you guys tell me what you think. Okay, so some viruses have this outer envelope and have these little proteins on the outside, but basically they're proteins and nucleic acids, and that's it. That's all they are. Maybe a couple enzymes thrown in there, and that's it. So let's go through what they are. So here, life, this is the definition: the condition that distinguishes animals from plants and inorganic matter is growth, reproduction, functional activity, and continual change preceding death. So, I don't know, let's see. Because, I mean, bacteria don't have continual change preceding death, but we consider them alive. And, for example, like chlamydia, they have to replicate, they don't make their own ATP, they have to use our ATP, and they're bacterial. It gets all real fuzzy. Okay, but I think it's cool to think about. So this is why they're over here. Now, after I've said it 10 times, now you get why they're over here in this weird no man's land. They're not on our phylogenetic tree of life because there is no life. They're not really considered living. So they belong over off the chart. So I'll talk a little bit about viroids and about prions and what those are, but mostly we're gonna focus on viruses. Okay. So here, naming viruses, I don't want you to memorize any of these, but I do think it's a good idea to just go like, oh, adenoviridae, coronaviridae, filoviridae, flaviviridae. Just go through a couple of these just so in your brain you can recognize a couple of them as you move through your science career. And we're gonna understand what this genome means. Later on, we're gonna understand what this capsid and morphology means. But here's SARS-CoV-2 is in this coronaviridae family. Influenza, so the flu, that's it. I'm sorry, go ahead. Do you have a question?
Yeah, there are single strands of DNA?
Yes, I'll tell you all about that when we do on Tuesday viral genetics. Yeah, super weird. Yeah, viruses get all kinds of weird. So here's influenza. So influenza is in the Orthomyxoviridae family. Influenza is what causes the flu. So influenza, we already know if your patient has the flu, they do not have a bacterial infection. It's not bacterial, it's viral. Okay, so we'll talk a little bit more about that. HPV, human papillomavirus. Have you guys heard of human papillomavirus? Human papillomavirus is HPV that can cause cervical cancer. It's a sexually transmitted infection, but it also causes warts, like plantar warts on the bottom of people's feet, is from HPV. Super weird. HPV is in the papilloviridae family. HIV, we've heard of human immunodeficiency virus, right? HIV. So we're gonna learn a lot about HIV. And so human immunodeficiency virus is in the retroviridae family. So I don't want you to memorize any of those, or even the ones I brought up. You don't have to know any families, just throwing it out there. That's how they're classified. So we don't really classify them as genus and species, like, because they're not really living. We kind of name them in a different way. We put them in a different family, sort of in a different way. So we name them kind of on their own little turf. Okay, so let's talk about the structure and function. So here's our size. I'm just gonna go on for a few more minutes. Hang in there with me. So I wanted to bring this back up again because I thought this video was so cool. No, don't want to log in. What did I do last time? Do you guys remember how I got? What did I do?
You scroll down.
Oh, yeah. Okay, perfect. Okay, let's play that again. There's no noise. Let's just play it from the beginning. So here's a grain of salt. Here's a human T cell. That's a human white blood cell. Here's a bacteria. Here are the viruses. So here is HIV surrounding a human white blood cell, a human T cell. That's what HIV, that's their tissue tropism, is a human white blood cell. Okay. So I always think that little visual is kind of cool. Where was I? Here. Okay. So they're ultra microscopic. 2,000 could fit into one bacterial cell. 2,000. 50 million could fit into one human cell. Really strange structures and shapes, but the reason I like to learn about viruses versus learning about bacteria is because there's kind of two main groups, and that's kind of it. So they're, in my mind, a little bit easier, and I'll show you why. We're not going to spend a lot of time talking about these. If you, like Dr. Duffy, for example, if you're looking ever to get in the lab, the bacteriophage lab, that is a very cool lab to work in, because these are viruses that infect bacteria, and they're looking at ways to manipulate bacteria using viruses, or maybe even as an antibiotic treatment or something like that. So that's a cool lab to always work in. But they're called bacteriophages, so I do want you to know what the name of them are, and I want you to know the general, you don't have to know anything about the structure other than that's what they look like. But they're called T4 lambda is that's just the example. You don't have to know T4 lambda. There are different types, but we use T4 lambda in the lab when you are looking at like bacterial viruses. So these only affect bacteria. That's kind of as far as we go with that one. Then I'll talk to you about what these other shapes are. Okay, so I'm going to draw it up here. So up there, it looks a little more complicated than it actually is. There are two types, basically. So on the outside, they look like this, but I'm just gonna draw it in lines so that I don't have to draw them all out. So just pretend these little loops go all the way around. So these loops go all the way around. These are proteins called capsids. So this is a polyhedral virus. Many sides. That's just a many-sided virus where the capsid forms this many-sided shape. And then there's another kind that's a helical virus. And just pretend those go all the way around and around and around. These are capsids. it's nice that viruses are less complicated, so we don't have to stress about them that much, right?
There's 2 layers of the capsids.
Yeah, so what the capsids do is surround the DNA or RNA. So they wrap. It's like a wrap. Yeah. Okay. So they're in 2 main shapes. This is the helical. So that's the 2 main ideas, is that the capsid can be out and form a box around the DNA or RNA, or they can form a snake-like structure and surround the DNA or RNA. So these are our 2 main ones, and then we move on to one more level. And the next level that we move up to is if they're enveloped or not. So these ones can be enveloped, or these ones can be enveloped. But there's 2 main ones, either or can be enveloped. So an envelope goes around the outside or around the outside of this one. They can be naked or enveloped. So that's viruses, that's it. So a naked virus can be a polyhedral virus or a helical virus. An enveloped virus can be a helical, a polyhedral, or the last one here is complex. And the reason I didn't include complex up on my 2 pictures is because that's this one. This is a complex virus. This is the bacteriophages, which we're gonna kind of just throw out there. So a complex virus is kind of any non-traditional shape, and there are some funky non-traditional shape viruses. We don't talk about those very often because those aren't really the ones that make us sick. These are the 2 main ones that make us sick. Well, there's 4. So there's 4 options. Polyhedral naked, polyhedral enveloped. Helical naked, helical enveloped. So there's 4 main types. Yeah? Okay. Okay, let's finish talking about capsids and then we'll be done. Okay, so capsids are repeated proteins. That's all they are, is the same protein repeated over and over and over and over and over. But don't forget, this is a 3-dimensional shape. So some have different sides, some have 20 sides, some have more sides or less sides, depending on the type of virus. We're not gonna memorize any of those. Just know that they're polyhedral, many-sided. So you'll see that term icosahedral sometimes, like they're 20-sided or something like that. Don't worry about the number of sides, we don't care. All we care is that they have many sides. So they're proteins, repeated proteins, the same protein repeated over and over and over and over that surrounds the either here in the polyhedral shape or here in the helical shape. So here in the helical shape is Ebola. So here it's the capsids are going round and round and round and round and round. Out here is an envelope. So the envelope is not always necessarily circular, it can be, or the envelope can be like this, around the outside. So not super important, it's either enveloped or it's not. Yeah?
And if it's not enveloped, it's naked.
Yes.
Sorry.
Yes. So here's the capsid. Sometimes they're called nucleocapsids. That means they surround the nucleic acids. Nucleocapsids, they're just capsids, proteins. Here's the bacteriophage, which we're not really gonna focus on very much. Very cool. 10 to the 31 bacteriophages on the planet, more than every other organism on Earth. So remember we said that about archaea, but we can say that about viruses, but we can't really say organisms. So they're the most frequent thing that's living that's not living on Earth. I wouldn't know how we say that, but there are a lot of them on Earth. More than you would ever imagine. Okay, so we'll start talking about the envelope on Tuesday. So today we're going to go through, let me think. For the quiz, let's not talk about viruses. Let's go up through archaea. Because it's Thursday, right? I am all mixed up. Okay, yes, so quiz all the way up through archaea. bacterial control, and archaea. Yeah?
Let's do the antibiotics for the
Antibiotics, all bacterial control, yeah. Awesome. Prevention, identification, treatment, all the vaccines, the terminology, broad spectrum, super infection, all that's in those slide set. Yeah. Okay, so one more thing really quickly. I'm going to have, let me bring up everybody. We're gonna do our note card, but really quickly also, I'm going to have, so here was from our last quiz. Lots of good results. If you were here 7, 6, 5, 4, or 3, I want you to stay in class with me after. I'm gonna send the group out who was up there. And not that I'm calling you out for any reason at all. The reason, some people are just busy and just didn't have time to study. Not a big deal. But we're gonna sit in class and we're just gonna chat, and I'm gonna figure out if there's a way I can help you, and I actually have some ideas, so we're going to get together. So anybody 7 or lower, also, if you got a 9 and you really want a 10, and you keep getting 9s, or you feel like you're getting like a B, but you really have to get an A in this class because you got a B, an A, and P, you have to get an A or a B in this class if you wanna go to nursing school. If you wanna go to nursing school, they want you to get an A in A and P and a B in micro, or an A in micro and a B in A and P. They don't want 2 Bs also. So that's something else to think about. So if you're stressing like, I have to get an A in this class, then stay after with me as well, and we'll do some tactical stuff. Okay, so let's do our note card, and then what I'm gonna have you guys do is go down and finish your lab and finish your lab reports with Dr. Rosevear, and then we will be down there to meet with you and look at all the lab reports together, look at all of our results together. But remember, full PPE because we're dealing with staff in a mask. Okay, so this is Methanobrevibacter ruminantium. This is a mouthful. Does anybody need a note card? Everybody's good. Okay, let's say it 3 times. Let's say it slowly. Methanobrevibacter ruminantium. 2 more times. Methanobrevibacter ruminantium. Last time. Methanobrevibacter ruminantium. You say that if you're asleep. Question. What's the gram?
Negative. Positive.
We can't gram. They don't have peptidoglycan, right? Yeah. Yeah, there you go.
So, are
I
we
should have
done?
led with that.
Yeah.
You're like, why? Why don't they have peptidoglycan? Now I'm confused.
So are we
Yes.
done with the bacteria
We're done with
part?
bacterial parts.
Okay, so this is, okay, this is just.
And what we're gonna do is, instead of collecting them and having Dr. Rosevear collect and then count them and then reorganize them and give them all back, he's just going to check that you guys were here. We're gonna do a quick walkthrough and make sure everybody's doing their note card, and you can just keep them. Does that sound good? Everybody's good to go? Okay, yeah, so you can keep your note card for today. Yeah, no. Yeah, no, I know first. So you will be able to stain it, but we don't really gram stain things that don't have peptidoglycan in that way. Yeah, we don't do like the traditional gram stain, because you can use like crystal violet to dye stuff, but we do a gram stain to look at positives versus negatives. We can't culture viruses in the lab, so the only thing we'd really be dyeing Okay, so there's strict anaerobes. They're coccobacillus, live in the rumen of ruminants. Ruminants are animals that have a rumen, that usually they're grazers. They produce methane from CO2, and they think maybe they're looking at the association of archaea with obesity, maybe dental plaque, maybe these different things that we're not really 100% sure, maybe in mice and other species, but we don't know. So if you are, oh, this was kind of cool, how much methane is effective versus carbon dioxide at trapping greenhouse gases. So that's something that's cool to look at. Okay, so if you were 7 or below, or if you want to stay, you're welcome to stay. If you are 8 or above, then go ahead with Dr. Rozier, and we're just gonna do some practical study talk.